In our previous video, the early part of Chapter 3, we were talking quite a bit about the lipid bilayer. And if you'll remember, this is the cell membrane that surrounds eukaryotic cells. This particular slide is a depiction showing you how the bilayer is actually called selectively permeable, and that's because some molecules are small enough to pass straight through those lipids, the phospholipids, readily without any extra help. It just has this concentration gradient of goes from high concentration to low concentration. And in this particular little depiction, the blue area on the screen is your interstitial or extracellular fluid. The more tan, salmon orangey color, whatever you want to call that, down below at the bottom of the screen, that is your intracellular fluid or your cytoplasm. Okay, so in this depiction we're going from a high concentration from the interstitial fluid down to a low concentration in the intracellular fluid. So there are fewer CO2 and O2 molecules, which is your example here. There's fewer of those inside the cell in the interstitial part, or excuse me, in the cytoplasmic part of the cell, in that tan area. And so there are more on the outside in the interstitial fluid, and we're going to go take all the CO2 and O2 from the outside and put it into the cell to balance it out. Again, we're striving for that homeostasis. The other depiction in this is where it's showing other molecules and ions. Sometimes they are too big to just readily slip right between those phospholipids. So they need some extra help. It's not a passive process. It's an act of being an active process where they're going to need some help by these proteins that are embedded in the lipid bilayer. And so if there are no proteins available that help the larger molecules go from the outside to the in, or inside out even, they are going to just basically bounce right back into the fluid from which they came. So in this depiction, they are coming from the interstitial fluid. They're trying to get to the interior of the cell, but there are no proteins to help them go through. There's no active process happening here, so they're slipping down. They're hitting the lipid bilayer, but they're too big, so they bounce right back. And then they remain in that lipid bilayer, or excuse me, in the interstitial fluid until some proteins can be found. So as I mentioned, there are two types of transport mechanisms, and this is in regard to materials, elements, molecules, ions, those kinds of things moving into and out of the cell. Okay, and so we've got the passive transport mechanism, which again is where the smaller molecules can move from an area of high concentration to an area of low concentration, whichever way in or out of the cell that may be. And it doesn't require any help. It's just passive. It just slips right through, and by way of that concentration gradient. That's what makes it's sort of like a slide. It goes from high to low, just as if you were on a slide. And they're small enough where they're going to slip in between your phospholipid layers. So you know you've got that double bilayer happening where you've got the hydrophilic heads facing outward and your hydrophobic tails facing inward. Well, if you can picture that, they're one on top of the other, so the heads are at the top and bottom, and they're all lined up. There's many of them, you know, all in a row. Well, there's a little bit of teeny tiny space between the two rows, and so the ions and the molecules and things can slip right between those. So they're slipping basically right between the edges of what the tails would be. So like they're the tail's outer edge. or slipping right down alongside the head, down alongside the tail, and into or out of the cell. So try to picture that. Again, there's that little bit of space. No energy is required in this, so you're just using that concentration gradient to go right in or out. Active transport, as I mentioned, is much more difficult. It usually goes against the concentration gradient, and it needs some protein molecules embedded in the lipid bilayer to help them get in and out of the cell. And it also needs some form of energy to give it that little bit of push. Because think about if you were in a river and you were downstream and all the waves are coming towards you. You've got that big wave, that big push of water coming towards you, and you're facing upstream. So all the water's coming towards you and you're trying to walk upstream. Well, you're going to need some energy to do that. You're going to need a little bit of a push to get started because it's such a strong push against you. And so that works the same way. These molecules are moving in or out of the cell, but across or against the concentration gradient, and it needs some energy to help it get started. And so that energy is in the form of ATP, which we've talked about before and we'll talk about again. But ATP, that little bit of energy gets it started. Okay, and so that is your active transport. Building on this idea of going from a high concentration to a low concentration, we need to discuss the diffusion rate. And this is basically how quickly a solute will diffuse across a membrane through a particular type of solution, depending on what we're talking about. In the previous slide, we were talking about actual cells and the cytoplasm and the interior of the cell and the extracellular fluid or interstitial fluid on the outside of the cell. But this can also relate to more tangible things that we can touch as humans. So in a couple of slides from now, you're going to see an example of some sugar solution in a baggie placed in water, and it's going to demonstrate some of these ideas. And so the diffusion rate will depend on these 5 factors: the size of the atom or molecule, the temperature of the solvent. So the temperature, the solvent will be what the ions are going to be placed in. Okay, so if we're talking at the cellular level, it'll be the temperature inside and outside of the cell, of the fluid. Okay, if we're talking about tangible things, it'll be whatever kind of solvent you've put in a glass beaker or whatever you're using. The steepness of the concentration gradient. So again, think of a slide. If the slide is really steep, if it's almost vertical, let's say it's little less than upright, so it's just, I don't know, like almost a 44-degree angle, we'll say. It's very steep is what my point is. You're going to slide down that slide super fast, right? And you're not going to need any energy at all. You're just going to slip and slide right in and out. So it's going to go very fast. The movement is extremely fast. If the slide is tilted downward, more horizontally, you're going to have have a little bit more effort going. The gradient is going to be stronger, and so it's not going to be necessarily faster, because it won't be. The gradient will now be slow, so the movement of the molecules is going to be quite slow. If it's flat, of course, it's not going to go anywhere. It may ever so slightly move, but it will take an incredibly long time, and more than likely not all the particles will move. So we have to have this balance. We don't want them to go too fast in a lot of cases, but we don't want them to not move at all. So we've got to have this nice appropriate amount of steepness that happens to get these molecules across that concentration gradient. Remember, the concentration gradient is moving from high concentration to low concentration. So high concentration is a whole bunch of solutes, and then ions, atoms, molecules, those kinds of things. Low concentration, there's very few of these solutes happening on the other side, and so that difference is going to make that pull or that gradient happen and the movement happen. Okay, so some of the molecules that are in the high side have to move down to the area where the solutes are quite few. And that is, again, we're going back to this homeostasis idea. We're getting this balance happening so that our body is in harmony, our cells are in harmony, whatever we're looking at tangibly with a bag of sugar put into some water, those kinds of things. It all has to gel, it all has to balance and become this harmonious thing. The electrical charge, or when an atom becomes a molecule, it's going to, remember, change electrons. It's either going to get rid of some electrons, it's going to attract electrons, it's going to share electrons, and once that starts happening, these electrons get a charge. Remember, they're either going to be positive, like in a sodium ion, or negative, like in a chlorine ion. Once the charge happens, we call it an ion. The charge is also going to help aid in that pull across this concentration gradient. And so think again about magnets, how they have that positive and negative ends, and when they're put together, they have sort of a bouncing effect, if you will, because there's a strong pull pulling them towards each other, but yet they're not quite compatible enough to really have that covalent bond where they're really secure and touching one another. We've got this ionic bond happening where they're super close, they've got a super strong pull towards each other, but they can't quite touch. And so that negative and positive pull is going to help the concentration gradient happen. And so let's say you have some sodium ions on the outside of a cell in the interstitial fluid, they're positive charge, and then we have some chlorine molecules on the interior of the cell, and they're all negatively charged. They are going to pull towards one another, and if there's small enough molecules, they're going to just immediately slip across that concentration gradient and be pulled towards one another. And so that charge is quite important. And then the pressure, or the turgor as it's sometimes called, pressure will also help. So think of a tube of frosting. If you're frosting a cake, you've got this tube and you're going to squeeze it, or even a tube of toothpaste. It's probably more relatable to everybody. You're going to take the cap off, of course, and then you're going at the end of the tube, you're going to squeeze it, and at the opening of the tube, some toothpaste is going to come out. Well, that's because you're putting pressure on it, and the more pressure or more tightly you squeeze it, the more forceful expulsion of that toothpaste will come out. And so pressure is quite necessary in a lot of these cases. If our slide, let's say, was a little bit more horizontally than we wanted to, and our concentration gradient wasn't quite such a strong pull, same with the electrical charge, let's say, was a weak one, any pressure that was instigated on that slide will help you slide down the slide faster, or get your body from one side to the other. So these are kinds of the same ideas. When molecules are moving across a concentration gradient, we have to have these factors to consider. We've already discussed diffusion quite a bit, but here are some definitions for you that you'll need to know. So, diffusion is the net movement of molecules or ions from a region of high concentration to a region of low concentration. And we've already talked about that in depth, how we think of a slide and you or molecules sliding down the slide. There's not any energy really required for that. Osmosis, I'm sure you've all heard of before. This is the diffusion of water molecules across the selectively permeable membrane between two fluids with different water concentrations. And we can translate this to also be at the cellular level. Sometimes we don't really think of water being in our bodies, even though we're primarily made of water. Sometimes it's a little bit of an abstract idea, but the selectively permeable membrane in most cases in your body that we're talking about is going to be your phospholipid bilayer that we all talk about, the cell membrane. And it's got some spaces between it so that water can slip right through, going back and forth easily. And this is what osmosis is. And it utilizes diffusion, so there's still going to be a high concentration and a low concentration involved. Okay, so this is going to be water molecules that are moving across the permeable membrane. And the fluids that they're talking about would be your extracellular or interstitial fluid on the outside of the cell, between all of your cells, or the cytoplasm within your cells. Okay, so that's what osmosis is talking about. Tonicity is the relative concentrations of solutes in fluids separated by a selectively permeable membrane. And so let's say you have a balloon and you've filled it with water, and it's sort of pliable when you squeeze it. You can squeeze it pretty well because there's nothing inside really that's hard or stronger than water to give it some rigidity to it. So if you have a bunch of solutes in the fluid, let's say you put a whole bunch of pebbles inside the balloon and you filled it almost all the way up. Let's say you filled it 3/4 of the way with pebbles. So now you've got a balloon filled with water and 3/4 of the way filled with pebbles. Well, some of that water will have probably spilled out just because of the space involved. But now if you were to squeeze the balloon, there's going to be a lot of resistance to it because the pebbles create this tonicity, this strength to the membrane, and that's what this is talking about here. So concentrations of solutes in the fluids, whether it's inside the cell or outside of the cell, and again they're separated by the selectively permeable membrane, which would be your phospholipid bilayer. And we'll talk more about these in the next few slides. This slide's going to illustrate the idea of osmosis and tonicity. So remember, we have the semi-permeable membrane that's straight in the middle between the two cups, both on the left and the right, and molecules that are too large cannot pass through the semi-permeable membrane, but water can. Water will move from a high concentration of water to a low concentration of water. Hypertonic solutions will have more solutes than hypotonic solutions. So if you're looking at the illustration on the left, the hypertonic solution has more solutes in it than the solution on the right, which makes it hypotonic. And then the isotonic solutions are what we get once the hyper and hypotonic solutions and the water moving from high concentration to low concentration even out. And once you even out everything over time, you get this isotonic solution happening. Now, even though it looks like the solutions are uneven, they are actually quite balanced. And so we'll talk about that. So if we go back to the illustrations on the left, the hypertonic solution begins with a cup of water, like you see, and a whole bunch of solutes are placed into that beaker. And you can see there's quite a lot of them. Some of those solutes will take water molecules and attach it to them. So the water molecules are going to attach to these other molecules, these solutes that have been placed in this beaker. And as you take some water molecules and attach them to solutes, the free water that's flowing has actually a lesser number of water molecules than when you first started. So and that's because some of those water molecules have attached to the solutes. So now that we have less water than what we started with on the hypertonic side, the left side here, the hypotonic solution is going to take water from the hypotonic side, move through that semi-permeable membrane over to the hypertonic side. So now what we have is a balance of the free water molecules. Okay, and so that's why it's showing you've got free. You've got a balance here. But what in reality it's going to look like, if you look to the figure on the right, the far right here in the isotonic sides, see how the left beaker has more water in it than the one on the right. Well, in actuality, the number of free water molecules are exactly the same on both sides. Okay, and that is because the water from the hypertonic side, that left side, some of that free water that we started with attached to the solutes, thereby lowering the water that we started with, which made the hypotonic side push water over to the hypertonic side, which is why when you look at the isotonic side, that left beaker has what looks like more water than the one on the right. But in actuality, because it had to move some over, it is balanced. And so going back to the figure on the left, when you have this hypertonic side originally, and you start with the free water, you put the solutes in it on both sides, you have more solutes in the hypertonic side than you do on the hypotonic side. When those free water molecules begin to attach to the solutes and you have less water than what you started with, the water on the hypotonic side is going to move through that semi-permeable membrane over to the hypertonic side because it now has a state of high concentration to low concentration. So that high concentration of water is now been sort of forced or created on the hypotonic side, when in actuality they both began with the same amount of water, but there were more solutes in that hypertonic side which were taking away some of those water molecules, thereby lowering that water, making a less concentration of water on the hypertonic side. And because we still have the original amount of water that we started with on the hypotonic side, that's going to be your high concentration of water, and that's why it's going to move down that concentration gradient through osmosis. Okay. At the same time this is happening, some solutes are moving back and forth across that permeable membrane if they are small enough. That's how it is in reality. In this particular example, the solutes haven't changed. Okay, so the only thing that's changed really here is the amount of water, which is why when you look at that isotonic state, you have more, what seems like more water on the left than on the right, but in reality they're exactly the same, as are the amount of solutes. Watch some of the videos that I have in Canvas that help illustrate this. There's some live experiments that have been done that I have videos of from Mr. Anderson, the teacher that I like. And then we will do some examples of this in lab this week as well. Okay, it's one of our fun labs, and it makes this point make sense a lot more. This concept's a little tricky to grasp, so we're going to show you another illustration, which I think this mimics what you'll do in lab, but it gives a nice visual of what is happening across the three scenarios. So in beaker A, we have a solution of 2% sucrose. So we have water and we have sucrose in this liquid in the beaker. Then we have a bag full of sucrose, which is again just another name for sugar, or a type of sugar. We have 2% sucrose in this bag. So we have the same amount of sucrose in the bag that we do in the water of the beaker. And so when we put the bag in the beaker, the solutions of the 2% sucrose in the bag and 2% sucrose in the beaker remain the same, so we have this nice balanced state. The water has no reason to move because it also has the same amount of water in the beaker and in the bag. So nothing happens, and we can call this an isotonic solution, or even that the bag is isotonic. Either way, you can say it either way. Now, beaker B, this is where it starts to get a little tricky. We have a beaker full of water that has a 10% amount of sucrose in it. Okay, so we've got 10% sucrose in this water in the beaker. We still have a bag of 2% sucrose. We put the bag in the solution and things start to happen. Because there is less sugar inside the bag than in the beaker, that means that there is more water in the bag because there's more room than in the beaker. Okay, because the sucrose in the beaker has 10% and it's taking up more water than the 2% sucrose taking up water in the bag. So when you put the bag in this solution, water has a need to move again to get that balance going. So because there's more water inside the bag, it's going to move from that high concentration of water in the bag to the low concentration of water in the beaker to get to an isotonic state. In doing so, when the water moves out of the bag, it causes the bag to shrink. And that's what this is showing. So now we've created an isotonic solution as far as water goes, but the bag is said to be hypotonic to the beaker. Okay, so the bag is hypotonic because it has less sugar than the beaker. So when we say hypotonic and hypertonic, we are referring to the solutes, that solid or molecular matter, okay, in the fluid. The hypotonic and hypertonic solutes are what drive the liquid to move. In this case, it's going to be water that's moving. And again, think of it in this picture. You have more water in this bag than you do in the water, or excuse me, in you do of the beaker, because the beaker has less room for water to be because it has more solutes in it. Okay, and then you have more room in the bag to fill up with water because you don't have as many solutes in it. You don't have as much sucrose in it. So the solutes determine hyper and hypotonic. So in this case, the sugar is less in the bag than in the water. So the bag is hypotonic to the beaker, which causes the water to move out of the bag, causing crenation, which just means shriveling or breaking. Okay, so crenation actually is shrinking or shriveling. You can think of it like that too. And so now let's look at the opposite scenario in beaker C. Again, we still have the 2% sugar in the bag, also filled with water. The beaker is just water. There is no solutes in it at all. So in this case, we have more solutes in the bag than we do in the beaker. That alone creates a hypertonic scenario, a hypertonic bag to the beaker. So when you put this bag in the water in the beaker, the water has a higher concentration in the beaker than it does in the bag. Because the bag has solutes, you can't fill up the entire bag with water because there's has to be some room for those solutes in it. In the beaker, it's just water. There's no solutes, so it can take up all the space there is. Okay, so when you put the bag in the water needs to go from that high concentration in the beaker to the low concentration of water in the bag. So the bag will fill up with water from the beaker, and if allowed, it could continue to fill up in some cases and burst. That's called lysis, Lysis. In this case, it's not bursting, it's just swelling. Okay, so a hypertonic solution will create the solute to expand. That's what's happening in beaker C. A hypotonic solution will cause the solute to shrink because it's losing water, or crenate, as it's called. And that's what's happening in beaker B. In beaker A, we're great. We have everything we're striving for. Even amount of solutes, even amount of water. That creates an isotonic solution or scenario. So hopefully this visual helps you get a little bit better idea of what's happening. And again, the water movement is because of osmosis. This little video slide is showing you examples of what we just talked about using an example of red blood cells. So the first one is an isotonic solution where the red blood cells stay that nice disc-shaped, nice completely round. Sometimes it's called a donut shape because it has that little bit of an indent in between or in the middle there. But they don't change in volume. They're just going to stay the same. Okay, same amount of solutes inside and out, same amount of liquid inside and out. Immersion in the hypertonic solution is going to cause those cells to shrivel or crenate because, again, we've got too much water inside the cell than outside, and so the water inside the cell is going to escape and make it shrink. And then the last one, the hypotonic solution, will cause the cells to swell up because there is less water inside the cell when it's placed in the solution than outside, and so the water will enter into the cell, making it swell up. And again, sometimes it can burst, called lysis. And then the last picture that you see is just a real-life depiction of these three scenarios. Next, we'll talk about transport mechanisms. There are two types, passive transport and active transport. Passive transport takes no energy hardly at all. It just sort of floats along and allows movement to just automatically happen in a sense. But passive transport will transport solutes or ions or molecules across a semipermeable membrane using osmosis and diffusion. Okay, and so sometimes the osmosis and diffusion can actually happen using membrane proteins, although it's not typical, but sometimes there are membrane proteins that the channels are just left open to the inside and outside permanently. And so molecules that can diffuse across the semi-permeable membrane will just slip right through these channels. They'll just slip right through, and there's no opening and closing or energy of any kind really to be put forth. Active transport, however, will use membrane proteins. These are usually larger molecules that cannot diffuse or use osmosis to get across. They need energy to give them that oomph, if you will. They will be going across the concentration gradient. And so they're going from low concentration to high concentration. And so here's that situation again where, remember before when I said you're standing in a river and you're downstream, but you're facing upstream and all this water's rushing towards you and you're walking upstream. Well, you're going to need energy to help you get upstream. Okay, and it's the same idea here. Ions and molecules will move across the concentration gradient in the membrane proteins using energy, and typically in the form of ATP. Okay, passive transport goes down the concentration gradient just like you would expect from high concentration to low concentration. The next few slides will illustrate these types of transport mechanisms. To finish out this chapter, we have just a few more transport mechanisms to discuss. They are called endocytosis and exocytosis, and you can probably glean from the word itself what this means. Endo meaning inward or interior, and exo meaning outside or exterior. Cytosis is a condition of a cell. And so we've got endocytosis and exocytosis. Endocytosis is where particles are moved to the inside of a cell. So a particle will come up to a cell membrane and push inward on it so that a pocket forms. As it does so, the cell membrane wraps around the particle that is pushing on it, and it forms an entire circle. That circle will be pinched off and released to the interior of the cell, where that particle can be carried to the proper organelle that can use it. You might hear the term invaginate. That just means that the cell membrane is moving inward and it's forming a pocket. Okay. Phagocytosis, you might hear about this. It's an example of endocytosis. It's used in the immune system, part of the white blood cell processes, and you might also hear of it from a single-celled organism standpoint, most notably the amoeba. The amoeba, as we've talked before, has cytosol in it that will push these on the cell wall and make these arms that extend out from the body, and it helps it to move, but it also helps it to surround particles. Oftentimes this is used in getting rid of waste. So an amoeba, it will get rid of waste, or it will take in nutrients of sorts. So the amoeba's arms will actually surround the waste particles and pull those particles into the interior of the cell. And so that's an example of an organism that uses phagocytosis. Pinocytosis you might also hear about. It's just another example of endocytosis, where particles are being taken into the cell that require extracellular fluid, or particles from the extracellular fluid taken into the interior of the cell. Exocytosis is the exact opposite. Particles are going to be moved to the outside of the cell. So most notably, these are waste materials in most cases. So the particle will sort of do the same thing. It will push on the cell membrane until it forms again another pocket, sort of on the opposite way, moving towards the outside, and then it will burst and the particles will be expelled into the extracellular fluid. So exocytosis is a means of getting rid of waste, and endocytosis is a means of taking in necessary particles or molecules that the cell, the interior of the cell, requires. It's usually organelles that require these materials, and so they're taken in so the organelles can carry out their function. And the next slide is a video example of these two types of transport mechanisms, and then the last slide is just an example of phagocytosis.
Cells also use energy to transfer materials in bulk, but this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
This is the example of phagocytosis I was telling you about, using an amoeba as an example. There's some sort of waste particle that the arms are being surrounding the particle, and they're going to be pulling that particle into the interior of the cell. That's called phagocytosis. It's a type of transport mechanism. And it's called cell eating, which is what phagocytosis means. That wraps up chapter 3.